Hello. So very good evening to the student who have joined. Can you let me know in the chat box if my voice is audible to you? Also, please let me know if you can see in my video and the class PPT. Let me know in the chat box if everything is fine. Is my voice audible? Okay. Thank you so much. We will wait till more students join and then we will start today's session. very good evening to the students who have joined. We will wait for 5 more minutes and let others join, and then we will start today's session. Meanwhile, I hope my voice is audible to you and you can see the PPT properly. So, if you have any problem, let me know in the chat box. So, at 6 p.m. We will wait. Okay. So, I think I can start with the introductions, and meanwhile when more students will join, I will just let them in to the class. So, yeah, very good evening to the students who have joined. My name is Mousumi Deb and I am the PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I am your TA for this NPTEL course of Medicinal Chemistry for this January 2024 session. Today it is our week 4 live session for this course of Medicinal Chemistry, and today we are going to solve the questions which are related to the week 5. So, every week we are going ahead with a week. So, last week we had in our third live session we had covered the topics which were in your week 4 course content, and now it is the week 5 which is live right now. And so, today I will discuss 20 questions related to your week 5 topics, okay, and these type of question will help you to answer your assignments, and also it will help you to understand the course better and to learn the topics of medicinal chemistry in more detail. So, during the session, if you have any doubt or any query, you can let me know in the chat box. Okay, the chat box is open always. I am just watching the chat box. So, if you have any doubt, any query, you can let me know in the chat box. Since I am recording this session for smooth streaming, I have just kept your microphones and videos off so that we have a smooth streaming for this class, and this video everything will be shared to you later. So, you do not need to worry if you have any kind of network issues, you do not need to worry about it. So, before starting the session, like every day I will sum up few points. So, so there will be a problem solving sessions every week for this course of medicinal chemistry, and these sessions will be on Tuesday, on every Tuesday from 6 p.m. to 8 p.m. and these sessions will be live on the Google Meet platform. And if any network disruption happens from my side, do not leave the class. Okay, I will join as soon as possible. So, please do not leave the class if there is a network issue from my side. And if there is a network issue from your side, you can join at any time into the class. Okay, you do not need to worry about it. You can join at any time to the class if you have any kind of problem for your network. And since I am recording the session, you do not need to worry about missing anything, because I will share this lecture PPT and this video also with you in the later. So, during the sessions you can post your queries and questions, everything into the chat box. Okay, for smooth recording I have just muted you all, but if you have any doubt, any query, anything, you can just write your doubts or queries in the chat box. I will check your questions, your doubts, and then I will answer these doubts and queries. So, you do not need to worry about it. And if still any doubt persists after class, you can drop an email to me. I will share my email ID with you at the end of the session. Like every day I share my email ID, and you guys mail me about your problems, your queries. So you are feel free to do that. So if you have any problem, any doubt regarding the course, the topics I am teaching, just feel free to reach me through the email ID. Also, there is a discussion forum about this course of medicinal chemistry, and you can post your doubts, your query into that discussion forum also. And this presentation and class recording, as I have mentioned before, I will share to you soon. So you don't need to worry about missing anything if there is a network issue from your side. But if there is a network issue from my side, please wait for some time, do not leave immediately. Okay, I will try to join as soon as possible. So as I have mentioned, today it is our week 4 live session, and right now the week 5 of this course of medicinal chemistry is live on the SWAM portal. So I think you have checked the course content, whatever it is in the week 5, and also there is an assignment. So today what we will do is we will discuss 20 MCQs regarding the week 5 course content, and these questions will help you to understand these topics better, and also it will help you to answer your for answering your weekly assignments. So whenever I am showing you some question, okay, try to answer these questions. Okay, you may answer this question correctly, may not answer this question correctly, but it does not matter if your answer is right or wrong, but answering is very important, because otherwise I won't understand if you are listening to me properly or if you are engaging into the class right now. Okay, so whenever I am showing you some question, please try to answer that question. Even if your answer is wrong, there is no marks given for that particular question, right, for answering the questions in the class. So you don't need to worry about it, but please answer the questions. Even if your answer is wrong, doesn't matter. Okay, I will show you the correct answer later on, and we will discuss that topic. So, okay, so please answer as soon as I am showing you some question. There will be 4 answers, A, B, C or D. You have to write with whatever you are thinking is the correct one into the chat box. Okay, I will check your answers, and then we will discuss that topic. So as I have mentioned, today we will discuss the week 5 course content. So whatever is in the week 5 course content, I have designed 20 questions regarding this topic. So what are these topics? These are nucleic acids as drug target, which is divided into part 1 and part 2. Then we have miscellaneous drug targets. Then we have tutorial of nucleic acids and related topics. Then we have mechanisms of biological chemistry, which is divided into part 1, part 2 and part 3. So all the questions will be from this week 5 topics. So I think I will start with the questions. Meanwhile, if more students will join, then I will just add them, okay, into the class. So the first question of today's session is, the following antibacterial agent was used in World War II. What is the name of this structure? The options are number A, erythromycin, number B, chloramphenicol, number C, proflavin, and number D, rifamycin. So what is the correct answer for this question? This is the structure given, so you have to identify which antibacterial agent is this. Is it erythromycin, is it chloramphenicol, is it proflavin, or it is rifamycin? What is the name of this structure? Somebody wants to join. Okay, so I will show you again the problem number 1. So the first problem of today's session is, the following antibacterial agent was used in World War II. What is the name of the structure? This is the structure which is given here. This is the structure. You have to identify what is the name of this antibacterial agent. Erythromycin, chloramphenicol, proflavin or rifamycin. I will give you some time. So try to answer it in the chat box. A, B, C or D, which of the following you think is the correct one? Let me know in the chat box, and then I will show you the correct answer and we will discuss it. will check the chat box, and after checking your answers I will show you the correct answer and we will discuss it. Okay, so I have one answer which is saying it is number A. Others also should try to answer this question. Even if your answer is wrong, it doesn't matter. Okay, I will give you 2 more minutes and then I will show you the answer and we will discuss. What is the name of this antibacterial agent? All of you please try to answer this question. Even if your answer is wrong, it doesn't matter. I am saying it all the time. But all of you answering the question is very important for all of you. Is it erythromycin? Is it chloramphenicol? Is it proflavin, or is it rifampicin? Let me check. Okay, so I have few answers. So I have number A, number B, number A, number C, number A. So most of you are thinking it is number A. So I have 3 answers, A, B and C. Okay, let me check and then let us discuss. So the correct answer will be number C, proflavin. So it is a topical antiseptic agent which is used in wound dressing and prevent infections. This is the structure of erythromycin. Now let me show each and every structure of this compound. So this is the structure of erythromycin. So erythromycin is a macrolide, okay, kind of antibacterial agent. It is used to treat certain infection caused by bacteria such as infection of the respiratory tract including bronchitis and pneumonia. This is the structure of chloramphenicol which is used to treat serious infections in different parts of our body. And this is the structure of rifampicin, which is a class of medications called antimicrobacterial. It works by killing the bacteria that causes infection. So these are the structures of these agents, and this is the structure for the proflavin, which is a topical antiseptic agent, and it was used in the World War II for wound dressing and prevent infections. Okay. So now I will move toward the next problem. Problem number 2 says, what is the mode of action for proflavin? Your options are number A, alkylating agents, number B, chain cutting agents, number C, antisense agents, and number D, intercalating agents. So for each and every class of your antibacterial agents, there is a method of or mode of action, or the method of action for that particular agent. So proflavin is a particular class of antibacterial agent. Now you have to identify which is the mode of what is the mode of action, or what is the class of this particular antibacterial agent proflavin. Is it alkylating agent, or is it chain cutting agent, or antisense agent, or intercalating agent? What is the correct answer for problem number 2? I will give you some time, think and write in the chat box. After some time, I will check your answer and I will show you what is the correct answer, and then we will discuss it like always. What is the mode of action for proflavin? Alkylating agent, chain cutting agent, antisense agent, or intercalating agent? In which category it comes? Let me check the chat box now. Okay, so I have again I have many answers like number C, number D, number A, number B. So I have, I think, A, B, C or D. Everybody has given one option each, so it is good. It's like we have variety of answers. So now I will show you what is the correct answer and then we will discuss it. Okay. So the correct answer for problem number 2 is proflavin is a intercalating agent. Okay. So drugs, first we need to understand what are the intercalating agents. So basically these are the drugs which acts on the DNA. Okay, and as you can see, this is the structure of proflavin, okay, which I have mentioned in the last slide also. If you see carefully in this structure, so we have in a center aromatic system, right, and at the edges we have this amine ions, okay, so this NH3 plus ions we have. So what happens, this kind of anti-intercalating agents, what they do is they pass into the sugar phosphate backbone and they break this backbones. Okay. So if you see, look at this bonds carefully, these aromatic ring, they will interact with throughout the van der Waals interaction with this base pairs, and this amine group, they will interact with the ionic interactions with this OH minus O minus of this particular DNA backbone. If I will show you carefully, so these are called the chemical mutagens. Okay, intercalating agents, they contain a flat planar structure that intercalate themselves into the double helix of DNA. This distorts the helical structure, and at replication a template that contains an intercalated agent will cause insertion of a random extra base. The base pair addition is complete after another round of replication, during which the intercalating agent is lost. For example, ethylene bromide, proflavin, acridine orange, these are very common examples of intercalating agents. So intercalating drugs are the compound that contain planar or heteroaromatic features which slip between the base pair layers of the DNA double helix. Some of these drugs prefer to approach the helix via major group, others prefer access via minor group. Once they are inserted between the nucleic acid base pairs, the aromatic heteroaromatic rings are held there by van der Waals interaction with the base pairs above and the below. Several intercalating drugs also contain ionized group which can interact with the charged phosphate groups of the DNA backbone, thus strengthening the interaction. Okay. So proflavin is an example of a group of antibacterial compounds called aminoacridines, which are used during the World War I and II to treat deep surface wounds. These drugs proved highly effective in preventing infection and reduced the number of fatalities resulting from wound infection. Proflavin is completely ionized at pH 7 and interacts directly with the bacterial DNA. The flat tricyclic ring intercalates between the DNA base pairs and interacts them by van der Waals forces, while the ammonium cations form ionic bond with the negatively charged phosphate groups of the sugar phosphate backbone. Once inserted, proflavin deforms the DNA double helix and prevents the normal functions of replication and transcription. So this is the So this is the method of action for this proflavin or any kind of intercalating agent. So remember, this is the proflavin is a very important intercalating agents, and these intercalating drugs works on DNA by adding them up into the sugar phosphate backbone, okay, and distorting the double helix structure of DNA. And other examples for this kind of intercalating agents are ethylene bromide, proflavin and acridine orange. These are the common examples. Okay. This flat rings or this tricycling ring interacts with the van der Waals interaction with this backbone and then with the DNA base pairs, and this ammonium cations, they interacts with the negatively charged phosphate group of this sugar phosphate backbone. Okay. And this is the example, and this is the method of action of proflavin. So now I will move toward the problem number 3. So if you have any doubt or any query, any problem, just let me know in the chat box. During the class, if you have any doubt or any query regarding any question, let me know. Then and there I will just repeat that question once more. Okay. So now moving forward, Problem number 3, it says that what is the molecular target for the Vinca alkaloid used as anti-cancer agents? The options are number A tubulin, number B they transport protein for serotonin, number C DNA, number D cell membrane. What is the molecular target for the Vinca alkaloid used as anti-cancer agents? Again I will give you some time. Read the question carefully, read the options carefully and let me know in the chat box A, B, C or D which of the following will be the correct answer for this question. What is the molecular target for Vinca alkaloid used as anti-cancer agent? Let me check the chat box now. Number A. So most of you are thinking it is number A, and I have answers like number A and number C. Okay, let me show what is the correct answer and then let us discuss about it. So the molecular target for the Vinca alkaloids used as anti-cancer agent is, very good, number A tubulin. So this is the structure of the Vinca alkaloid. Okay, this is a natural product which is used as an anti-cancer agent. So generally during normal mitosis or during the metaphase, okay, so there is a chromosome and spindle formation happens, okay, and this spindle formation, if you look at carefully, there is a thing which is called the tubulin. So this is the tubulin molecule which are stacked in to form the mitotic spindle, and this by forming this spindle we have this chromosome division and we have this metaphase to anaphase, okay. This is a normal mitosis during, but when there is a Vinca alkaloid, this is used as an anti-cancer agent. So what happens? This kind of Vinca alkaloid, they this is the microtubule, and this microtubule is formed with this free tubulin dimers. Okay, so Vinca alkaloid, what they do is they bind with this free tubulin dimers. Okay, and when they are bound, these are, and then Vinca bound tubulin dimers are formed. And if this Vinca alkaloid are bound with this free tubulin dimer, and this dimers Vinca bound tubulin dimers are formed, what happen? The paracrystalline aggregates are formed with this particular Vinca bound tubulin dimer, and this tubulin formation ruptures. Okay, tubulin molecules fail to polymerize now in presence of this Vinca alkaloid, and this metaphase to anaphase, this format transport, this phase change does not happen, and what happened? Dissolution of the mitotic spindle leads to the cell death. So this is one method by which this cancer treatment can happen. So we all know cancer is due to the uncontrolled cell growth, we can say. Okay, so this particular Vinca alkaloid are used in case of cancer cells where they bind with this free tubulin dimers. So microtubule formation with this particular Vinca bound tubulin dimers is not possible, and then this dissolution of this mitotic spindle leads to the cell death of this cancer cell. Okay. So, this is the method of action for these benzoalkaloids, and this tubulin is the molecular target which for this particular anti-cancer agent. Okay. So, now moving forward, problem number 4, it says that which DNA base interacts with the anti-cancer intercalating agent dactinomycin. Your options are number A adenine, number B thymine, number C cytosine, and number D guanine. So, which DNA base interacts with the anti-cancer intercalating agent dactinomycin? Adenine, thymine, cytosine or guanine? Again I will repeat the problem number 4. It says which DNA base interacts with the anti-cancer intercalating agent dactinomycin. Your options are number A adenine, number B thymine, number C cytosine and number D guanine. I will give you 2 more minutes and then I will check your answers and we will discuss this question. All of you please try to answer these questions in the chat box. Even if your answer is wrong, it does not matter, since we are not giving you marks on this answering in the class, but this will only help you for understanding the course better and for your assignment purpose also. Now, let me check the chat box and then we will discuss this question. Okay. Okay, so I have received the answers number D, number D. So, most of you are thinking it is number D, and then we have number A and number C as answers also. Okay, fine. Let me check and then we will discuss it out. So, the correct answer will be very good, number D, guanine. So, guanine is the DNA base that interacts with the anti-cancer intercalating agent dactinomycin. So, this is the another example of the intercalating agent. So, last intercalating agent what we discussed was the proflavin. So, if you think about the structure of the proflavin and then if you check the structure of this particular dactinomycin, so you will see there is also a tricyclic aromatic structure here. Okay, just like proflavin. So, this is a particular feature of the intercalating agents. So, another example of this intercalating agent is dactinomycin, and it is called the actinomycin D. previously. It is a naturally occurring antibiotic that was first isolated from the streptomycin parvovirus in 1953, and it has shown that it have an effect as anti-cancer agent in childhood cancers. If you look at the structure of dactinomycin, it has a planar phenoxazone system, and it also has a peptide over here, which is a cyclic peptide. So, we say this structure, the center core, it is called a phenoxazone, okay, phenoxazone system, and we have peptide, a cyclic peptide here at the periphery, okay. And the drug approaches DNA via the major group of the double helix, and after it intercalates, the planar tricyclic system gets between two base pair, as we have seen previously. The charged amino group attached to the sugar is also important, and it forms ionic bond with the negatively charged phosphate groups of DNA backbone. So, this is the feature we have discussed for the proflavin also, that this is the thing is same, that the planar tricyclic system intercalates between the base pairs, okay, and the amine group, which are the charged amine groups, they are they bonded with the negatively charged phosphate groups of this DNA phosphate backbone. And this dactinomycin, it approaches the DNA via the major group of the double helix. It appears to favor the interaction with the guanine cytosine base pair, that is the GC base pairs, and in particular between two adjacent guanine bases in the alternative strand of helix. So, if you have GC interaction and you have two Gs between, it prefer to bind this type of DNA base pairs, and the molecule is further held in a position of hydrogen bond interaction between the nucleic acid bases and cyclic pentapeptide, as we looked earlier. The 2-amino group of the guanine plays a particular important role in this interaction. The resulting bound complex is very stable, and it prevents the unwinding of the double helix. So, this in turn prevents the DNA dependent RNA polymerase from catalyzing the synthesis of mRNA, and thus it prevents the transcription. So, the effect of dactinomycin is actively dividing cell is that once it gets in, it goes inside and does intercalate, then after it intercalates, this form an extremely stable bond with the DNA, and after this complex is formed, because this complex is so stable, it prevents the unwinding of the double helix, and if the unwinding of double helix does not happen, as we all know from previous lectures, that the single strand DNA is not produced, which is going to transfer the information to mRNA, and so there will be the transcription is inhibited, and as a consequence, if the transcription is inhibited, the cell will stop replicating and stop dividing, okay. So, the question can be given in this matter also, that the in which process of the cell division the dactinomycin it inhibits. So, remember, it inhibits this particular this transcription process, okay, and it does not because we don't have this unwinding of double helix does not happen. So, we don't have this transcription process, and this transfer of information to mRNA does not happen, and the cell it will stop replicating and dividing if there is a no proper transcription process happens. So, this is the particular method of action of this anti-cancer intercalating agent dactinomycin, and the DNA base pair which it prefers most is the guanine, okay. So, now I will move toward the next problem. Problem number 5 says, for which phase of cell cycle the anthracycline anti-cancer agents are toxic, okay. For which phase of cell cycle the anthracycline anti-cancer agents are toxic? Your options are number A S, number B M, number C G1, and number D G2. Again, I will repeat the problem number 5. It says that for which phase of cell cycle the anthracycline anti-cancer agents are toxic? Your options are number A S, number B M, number C G1, and number D G2. Again, I will give you some time. Think and write in the chat box which of the following options A, B, C or D you think is the correct one, and then we will discuss this. Again I will repeat the problem number 5, for which phase of cell cycle the anthracycline anti-cancer agents are toxic. Number A S, number B M, number C G1 and number D G2. Let me check the chat box now. Okay, so I have answers for this question also. B A, number B, number C, number B, doxorubicin S phase of the cycle. Okay, let me check which answer is correct and which is not. So the correct answer will be, very good, number A S. So S phase of cell cycle is for S cell of cell cycle that anthracycline anti-cancer agents are toxic. So anthracyclic so this is one class of antibiotic agent, the anthracyclines. So for example, there are doxorubicin and your daunorubicin. So anthracyclines are the tetracycline class. So there is a tetracycline rings with the sugar daunomycin, and they are DNA intercalating agent that block the synthesis of DNA and RNA. These agents are primarily toxic during the S phase of the cell cycle. Doxorubicin is probably the most important anti-cancer drug available because it is the relatively broad spectrum of activity. So as you can see, there is a normal DNA. Okay, so from this normal DNA we have this DNA synthesis happens, but whenever there is this agent doxorubicin, so it will intercalate between the double helix again, and there is a formation of DNA doxorubicin complex, and because of this DNA synthesis does not happen. And there is in the cell cycle, if you remember, or if you have read the cell cycle, if you have not, we will discuss the cell cycle later on also in our course. So in the cell cycle there are 4 phases, right? We have this G1 phase, then we have this S phase, we have G2 phase, and then we have the M phase also. Now in the S phase the DNA synthesis happens, okay, and this anthracycline class of anti-cancer agents, they target this S phase of the cell cycle, and since they are the intercalates between the double helix structure of the DNA synthesis does not happen, okay, and we have the cell death. So that is how they exert their anti-cancer property. So now I will move toward the next problem. Okay, I will just discuss it also. Intercalation prevents the normal action of enzyme called topoisomerase 2. This is an enzyme that is crucial to replication and mitosis. Mechanism of topoisomerase 2 involve the formation of a DNA enzyme complex, and where the enzyme covalently linked to the DNA. When doxorubicin is intercalated into the DNA, it stabilizes this DNA enzyme complex and stalls the process. Okay. So this was the mechanism of action of anthracycline anti-cancer agents, and the cell cycle phase for which they are toxic is the S phase, okay, and they prevent the normal action of the enzyme called topoisomerase 2. This is also important, remember this point also. So now moving forward, problem number 6, it says that the following structure is used for the treatment of brain tumors. What is the structure called? This is structure, you have to identify the name of this structure. Your options are number A carmustine, number B lomustine, number C streptozotocin, and number D cyclophosphamide. What is the name of this structure? This is used for the treatment of the brain tumors. Identify the correct name of this structure. Is it carmustine or is it lomustine, then streptozotocin, and then cyclophosphamide. What is the name of this structure? Let me check the chat box. Okay, so I have number A, B and C. So I have, I think, number A, B, C, various answers. So let me check which of the following is the correct one, and then let us discuss about it. So the correct answer for this question will be number B, lomustane. So this structure of the name of the structure is lomustane, and now let me show you. So this is an alkylating nitrosourea compound which is used in the chemotherapy. So lomustane, it is a it is an alkylating nitrosourea compound used in chemotherapy. It is closely related to the semustane and is in the same family of the streptozocin. It is a highly lipid soluble drug and it also crosses the blood brain barrier. This property makes it ideal for treating the brain tumor, which is its primary use, although it is also used to treat the Hodgkin lymphoma as the second line option. Okay, so this compound, it is very much lipid soluble, and since it is highly lipid soluble, it able to cross the blood brain barrier. We will discuss about this blood brain barrier and everything later in our course. Okay, and one thing you need to know is blood brain barrier is the barrier between the so that the most of the drugs which cannot pass through our brains. Okay, but for treatment of brain tumor, the drugs must cross this blood brain barrier, and for this kind of drugs they need to be highly lipid soluble. Okay, and that is why this drug is suitable for the treatment of the brain tumor. This is the structure for the carmustane. It works by slowing or stopping the stopping the cancer cells. Okay, so if you see the structure of the carmustane and lomustane are almost the same, the difference is only in this portion. Okay, so remember, if there is a cyclic structure, this cyclohexane ring, so we will have the name is the lomustane, and if there is a chain chlorine containing then this is the carmustane. Okay, and all of these are the same family of the streptozotocin. It is an antibiotic that produces the pancreatic islet beta cell destruction. Okay, and then we have this cyclophosphoramide, which is used to treat the cancer of the ovaries, breast, blood and lymph system. Okay, and these compounds are the nitroso and alkylating compound. These agents are the alkylating anti-cancer agents. Okay. So now next problem. Problem number 7 say the major intercalating species in bleomycin is number A, thiazole ring, number B, imidazole ring, number C, bis-thiazole ring and number D, indole ring. So what is the major intercalating species in bleomycin? Thiazole ring, imidazole ring, bis-thiazole ring or indole ring? Let me know in the chat box. A, B, C or D, which of the following is the correct one? The major intercalating species in bleomycin is thiazole ring, imidazole ring, bis-thiazole ring or indole ring. The major intercalating species in bleomycin is thiazole ring, imidazole ring, bisthiazole ring or indole ring. Let me check the chat box and then I will show you the answer and we will discuss this. Okay. So, I have few answers. Number A, all of you think it is the number A. Okay. Let me check if number A is the correct one and then we can discuss it. Okay. So, the correct answer will be number C, bisthiazole ring. Now, this is a little bit tricky. So, let me show you the answers first and then let us discuss it. So, this is the structure of the bleomycin. So, this portion it is called the thiazole, right. But if you can see, look at carefully, we have two thiazole ring joined to each other. This whole system it is called the bisthiazole system and the major intercalating species of bleomycin is this bisthiazole system. Their structure includes the bisthiazole system which intercalates with DNA. So, now the major intercalating species of the bleomycin is this bisthiazole ring. One ring is the thiazole, but since there is a two thiazole joined to each other, this is called the bisthiazole system and this bisthiazole system is the major intercalating species of the bleomycin. Okay. So, bleomycin, the mechanism of action is it acts through the binding to the DNA which results in single and the double strand breaks following the free radical formation and inhibition of the DNA synthesis. The DNA fragmentation is due to the oxidation of a DNA bleomycin iron complex and it leads to the chromosomal aberrations. Cell cycle specific drug that causes accumulation of cells in the G2 phase. Once the structure becomes intercalated, the nitrogen the nitrogen atoms of the primary amines, the pyrimidine ring and the imidazole ring chelate an ion that is ferrous ion which then interacts with oxygen and is oxidized with the ferric ion. Now, during this process oxygen picks up an electron and forms a superoxide and superoxide can subsequently get converted to the hydrogen peroxide which then eventually forms the hydroxyl radical. These together are known as a reactive oxygen species and the cell is very efficient in getting rid of some of this reactive oxygen species. So, now however, since the generation of reactive oxygen species occur near the DNA strands, this can result in the DNA strands being cut particular between the purine and pyrimidine nucleotides. Bleomycin also appears to prevent the enzyme DNA ligase from repairing the damage caused whenever there is a damage that occurs in DNA and there are number of enzymes that come and help repairing the damage that is caused. Okay. So, the major intercalating species of bleomycin is, remember, this is the bisthiazole ring system. And you do not need to memorize this complicated structures of this kind of molecule, but the features, the structural features which are important for their mechanism of action you need to remember. Okay. So, now moving forward, problem number 8 says find out the incorrect statement among the following. Number A, it says that moxifloxacin is a quinolone class of antibiotic drug. Number B, method of action for quinolones is the inhibition of DNA guidase. Number C, doxycycline works by stopping the DNA replication. And number D, tetracyclines prevent the binding of tRNA with the A site. So, what is the incorrect statement or which of the following statement is incorrect? Again I repeat, number A, moxifloxacin is a quinolone class of antibiotic drug. Number B, method of action of quinolones is the inhibition of DNA guidase. Number C, doxycycline works by stopping the DNA replication and number D, tetracyclines prevent the binding of tRNA with the A site. Read all the options carefully and let me know which of the following is the incorrect one. I will give you some time for this question, but read all the options carefully and let me know in the chat box which of the following is the incorrect statement. Find out the incorrect statement among the following. Moxifloxacin is a quinolone class of antibiotic drug. Number A. Number B. Method of action of quinolones is the inhibition of DNA guidase. Number C. Doxycycline works by stopping DNA replication. And number D. Tetracyclines prevent the binding of tRNA with the A site. Let me check the chat box now. Okay. Number A, number C, number A. So, I have two answers, number A and number C, and so let us check if number A is incorrect or number C is incorrect for this question. So, the incorrect statement is number C, doxycycline works by stopping DNA replication. It is the incorrect statement. This is the structure of the doxycycline. So, first let us discuss about the mechanism of action of your quinolone class of antibiotics. So, the mechanism of action, they block the bacterial DNA synthesis by inhibiting the bacterial topoisomerase 2 or DNA guidase and topoisomerase 4. Inhibition of DNA guidase prevents the relaxation of positively supercoiled DNA, and that is required for normal transcription and replication. Inhibition of topoisomerase 4 probably interferes with the separation of replicated chromosomal DNA into the respective daughter cells during the cell division. Okay. So, the example of quinolone class of antibiotics are nalidixic acid, perfloxacin, ciprofloxacin, levofloxacin, moxifloxacin, ciprofloxacin, and so on. So, this is the common structure of this fluoroquinolone structure, where we have this quinolone ring here, okay, present, and there is a fluorine also present into this 6th position of this quinolone ring. Okay. So, this is the mechanism of action for quinolone class of antibiotic is the inhibition of DNA guidase. So, so your option number A and option number B are the correct one. Okay. So, now let us discuss about the tetracyclines. So, the tetracyclines, they prevent the tRNA, they actually prevent this particular translation process, not the DNA replication process. Okay. Doxycycline is the group of compound which is known as the tetracycline tetracyclines. Okay. So, these tetracyclines, they work, how they work, they actually basically they prevent the binding of tRNA with the A site. So, this is the this is the ribosome, this is the 50th subunit, and this is the 30th subunit of the ribosome, and there are two sides, one is the P side and one is the A side. Okay. This one is A side, the tRNA binds, okay, and this is the called the transfer side. So, what happens, this tetracycline, they bind to this A side of the tRNA, which is the binding with the tRNA of the A. So, even the binding of tRNA with A site, because they bind to this A site, the tRNA now cannot bind, and if the tRNA cannot bind, this particular translation or protein synthesis process is inhibited, and that is how they exert their antibacterial property. So, remember, doxycycline does not work by stopping the DNA replication, they work by binding to the A side of this ribosome, 30th ribosome, so that the tRNA now cannot bind and the protein synthesis is inhibited. Okay. So, doxycycline or tetracycline, they work by stopping the protein synthesis, not the DNA replication. So, statement number 3 C is the incorrect one, and all other statements A, B and D are the correct ones. Okay. So, problem number 9, it says that etoposide is number A, intercalating topoisomerase poison, number B, non-intercalating topoisomerase poison, number C, intercalating topoisomerase inhibitor, and number D, non-intercalating topoisomerase inhibitor. So, now poison and inhibitor, these two are they may seem to be same, but they are not the same. These terms are there are differences of this term poison and inhibitors. Okay. So, this is the structure of the etoposide. So, now you have to identify which of the following is the correct one. Etoposide is a number A, intercalating topoisomerase poison, number B, non-intercalating topoisomerase poison, number C intercalating topoisomerase inhibitor, number D non-intercalating topoisomerase inhibitor. Which of the following is the correct one, you have to identify. Etoposide is, this is the structure of etoposide. Now identify which of the following is the correct one. It is the intercalating topoisomerase poison, or is it a non-intercalating topoisomerase poison, or is it number C intercalating topoisomerase inhibitor, or number D non-intercalating topoisomerase inhibitor. Let me check the chat box. Number D, number C. So, number C and number D. So, I have received these two answers till now. These two options are given as answers, number C and number D. So, let me check which of the following is the correct one. So, the correct answer is, it is number B. It is a non-intercalating topoisomerase poison. Okay. So, now let us look at some non-intercalating topoisomerase poison. The anti-cancer agent etoposide and teniposide, they belong to a class of the compounds known as the podophylotoxins, and these are the semi-synthetic derivatives of epipodophylotoxin as an isomer of the naturally occurring agent called the podophylotoxin. Now, both of these agents act as a topoisomerase poisons. DNA strand breakage by this class of molecules is thought to occur by a free radical process that involves 4' phenolic group, that 4' phenolic group, this one. Okay. And what happens, this is proposed to happen that 4' phenolic group forms a semiquinone radical which generates the again reactive oxygen species, and evidence of this comes from, if you remove this and if you remove this, you will able to derivatize this oxygen into the methyl, and then the compound becomes inactive. The presence of this glucoside sugar moiety is also increase the ability of this molecule to induce the breaks. So, what is the mechanism of action of this kind of topoisomerase poisons is, there is a phenolic OH at the 4' right, this one. So, this 4' phenolic OH forms a phenoxide radicals, and this phenoxide radicals forms a semi-quinone structures. Okay. And because of this quinone structure is formed, this is this poisons the particular topoisomerase enzyme. But so, what is the proof of this particular thing, that if you derivatize this OH, this let us say you are derivatizing this OH as OMe, okay, then this radical formation cannot happen, and then this compound will become inactive. This presence of this portion is also important, and this also increase the ability of the molecule to induce the breaks into the particular DNA. Okay. So, remember etoposide, teniposide, these are the non-intercalating topoisomerase poison. So, now problem number 10, it says which reaction is involved in the method of action of calcisamine. Your options are number A Balli-Silman reaction, number B Suzuki coupling reaction, number C Diels-Alder reaction, and number D Bergman cyclization. which is involved in the method of action of calcisamycin? Balli-Silman reaction, Suzuki coupling reaction, Diels-Alder reaction or Bergman cyclization reaction? Let me know in the chat box A, B, C or D, which of the following is the correct answer. Which reaction is involved in the method of action of calcisamycin? Let me check the chat box and then I will show you the answer. Okay, number D, number B. So I have two options as answers. Let me check and then let us discuss about it. So the correct answer will be number D. Bergman cyclization is the method of action for the calcisamycin. So this is the mechanism of Bergman cyclization. So as you can see, this is called the indiene. Indiene means there will be a triple bond followed by a double bond and then we will have a triple bond. So if you have heat or H nu, heat or light condition, photochemical or under thermal or photochemical condition, what happens, this triple bond they form radicals, okay, and we have this 1,4-benzene diol radical, or it is called 1,4-benzenoid or parabenzene diradical, and this diradical they take up there happens, there is a cycloaromatization happens, okay, and because of the cycloaromatization we have this kind of formation. So this is the Bergman cyclization mechanism. Now let us see the method of action for calcisamycin. So this is the structure of the calcisamycin, okay, and this there is a sugar moiety here and we have this N-diene structure here presence and we have this sulfide bonds present here. So first what happened, nucleophile they attacks here and disulfide bond breaks, okay, and we have a free thiol group. This sulfur acts as a nucleophile and attacks on this position or this carbon, and we have this kind of your racemization or tautomerization we have here, and then what happened, this Bergman cyclization happen in this indiene portion, okay, and due to this Bergman cyclization what happened, we have this benzene diradicals and this and if there is a DNA diradical form and this DNA double strand cleavage happens because in presence of oxygen, okay. So the method of action, the reaction which is involved in the method of action of calcisamycin is the Bergman cyclization reaction. So now next question, problem number 11 has said that camptothecin works as number A topoisomerase 1 inhibitor, number B topoisomerase 2 inhibitor, number C DNA guidase inhibitor, number D DNA polymerase inhibitor. Camptothecin CPT, camptothecin, the short form of camptothecin is CPT. So it works as a topoisomerase 1 inhibitor, topoisomerase 2 inhibitor, DNA guidase inhibitor or DNA polymerase inhibitor? Again I will give you some time. A, B, C or D, which of the following is the correct answer? Let me know in the chat box. After your trial, I will show you the answer and we will discuss about the camptothecin. So, camptothecin works as a topoisomerase 1 inhibitor, topoisomerase 2 inhibitor, DNA guidase inhibitor or DNA polymerase inhibitor. I will give you one more minute and then I will show you the answer and we will discuss about it. Let me check the chat box now. Okay, so number D. So, all of you, it is the number D. Okay, let me check what is the correct answer and then let us discuss about it. So, the correct answer will be number A, topoisomerase 1 inhibitor. So, camptothecin or this is the CPT, it is a plant alkaloid. Okay. So, this is the active form which is a camptothecin which is a lactone form, and if there is a base present, so this lactone will be formed as a carboxylate. So, this is a camptothecin which is the inactive form. Okay. So, it is a plant alkaloids and CPT is the most established inhibitor of DNA topoisomerase 1. So, CPT or camptothecin is a it is a very important, you have to remember that it is the most established inhibitor of human DNA topoisomerase 1 enzyme. Okay. And this is this particular molecule is pharmacologically unique because topoisomerase 1 is the only target of this drug. So, this molecule is very important, that because this topoisomerase 1 is the only target of this drug and CPT binds neither to topoisomerase 1 alone or not to the DNA alone, but only to topoisomerase 1 DNA cleavable complex. Okay, so these points are very important, that camptothecin they bind it does not bind to topoisomerase 1 enzyme or DNA alone, but it binds to the topoisomerase 1 DNA cleavable complex. So, if you look at this carefully, there is a cyclic quinolone ring here present, and then this is a there is a macrocyclic kind of structure of this. There are various rings which are fused, heterocyclic structure. There are various rings which are fused to each other. Okay. So, it is a very important molecule is camptothecin. Camptothecin is a natural product that was extracted from the Chinese bush Camptothecia acuminata in 1966. Camptothecin stabilizes the cleavable complex that is formed between DNA and the enzyme topoisomerase 1. So, by doing this, what happens that it results in single strand breaks into that accumulate in DNA. So, you may remember the topoisomerase 1 and 2 both nick the DNA for their function. The difference between topoisomerase 1 and topoisomerase 2 is that topoisomerase 2 cleaves only one strand of DNA, whereas topoisomerase 1 cleaves only one strand of DNA, and the topoisomerase 2 cleaves the both strands. And since the complex between DNA and enzyme topoisomerase 1 is stabilized by has stabilized the single strand break persists, it does not therefore accumulates in the DNA, resulting in the inhibition of proliferation. These single strand breaks can be repaired if the drug departs, but if replication is taking place when the drug enzyme DNA complex is present, an irreversible double strand break takes place, which then leads to the cell death. Semisynthetic analogues of camptothecin have been developed as clinically useful anti-cancer agents. Okay. So, remember camptothecin is the most established inhibitor of DNA topoisomerase 1. Okay. So, now problem number 12, it says that which of the following statement is true regarding the antibodies. Your options are: number A, T cells are associated with the humoral immunity. Number B, B cells are associated with the cellular immunity. Number C, B lymphocytes are present in the bone marrow, and number D, B lymphocytes generated by the thymus. Which of the following statement is true regarding the antibodies? Number A, T cells are associated with the humoral immunity. Number B, B cells are associated with the cellular immunity. Number C, B lymphocytes are present in the bone marrow, and number D, B lymphocytes are generated by the thymus. Which of the following statement is true regarding antibodies? Let me check the chat box. Okay, number A is the most given answer. Let me check if the number A is correct one, and then let us discuss about it. Okay, so the correct answer or the statement which is true regarding antibodies is number C, B lymphocytes are present in bone marrow. This statement is true. So, first discuss about the difference between the B cells and T cells. So, B cells, they attack the inhibitors which outside the cell, and T lymphocytes or T cells, they attack the infected cells itself. Okay. So, the differences between the B lymphocyte and T lymphocyte is that B lymphocyte, the origination and the maturation, they takes place in the bone marrow. Okay. And the T lymphocyte, the origination in the bone marrow, but the maturation in the thymus. They differentiate into the plasma cell, the B lymphocyte. T lymphocyte differentiate into the plasma cell or memory cell or cytotoxic T cell. This B lymphocytes, they involved in the humoral immunity, but T lymphocytes, they are involved in the cell mediated or cellular immunity. The B lymphocyte they produce the antibodies, and the T lymphocyte they help the B lymphocytes to produce antibodies, and B lymphocyte they produce the antibody, but T lymphocyte they help B lymphocytes. Okay. So, remember B lymphocyte the origination and maturation takes place in bone marrow. So, for T lymphocyte the origination happens in bone marrow, but the maturation takes place in the thymus. For B lymphocyte they differentiate into the plasma cell, T lymphocyte differentiate into the plasma cell or memory cell or cytotoxic T cells. B lymphocyte involved in the humoral immunity, T lymphocyte involved in the cellular mediated immunity. B lymphocyte produce the antibodies, but T lymphocyte they help the B lymphocytes to produce the antibodies. Okay. So, this is the differences between the B lymphocytes and T lymphocytes. So, now next problem. Problem number 13 says how oxalidenone works. Number A, it inhibits the protein synthesis by binding to 30S ribosome. Number B, it inhibits the protein synthesis by binding to 50S ribosome. Number C, it unwinds the DNA double helix, and number D, it inhibits the DNA guidase enzyme. So, oxalininones are another class of antibacterial agents, okay, and you have to identify what is the mechanism of action for this particular class of antibacterial agent called oxalininone. Number A, inhibition of the protein synthesis by binding to 30S ribosome. Number B, inhibition of the protein synthesis by the binding to the 50S ribosome. Number C, unwinding of the DNA double helix, or number D, inhibition of the DNA guidase enzyme. Let me know in the chat box A, B, C or D which of the following options is the correct one for this problem number 13. Let me check the chat box. Okay, number B, number A. Okay, let me show you the answer and then let us discuss about this question. Okay, so the correct answer means the mechanism of action for oxalininone is number B. It inhibits the protein synthesis by binding to the 50S ribosome. So, oxalininone, linazoloid, this radicoloid or torzaloid. Linazoloid is the totally synthetic antibiotic active against the gram-positive organism including the streptococci, cephalococci, enterococci and gram-positive enterococci and gram-positive rods such as corneobacterium and listeria mitogenes. The mechanism of action is it inhibits the bacterial protein synthesis by binding to the 50S ribosomal subunit near the interface of 30S, and thus the formation of initiation complex is prevented. This is the core structure of the oxalininone. This heterocyclic ring which is present in blue, it is known as the oxalininone. Okay, and this is so there is a structure number B. So this is the core structure. We have a auxiliary ion core here, we have a side chain in red, and then we have this ring in pinkish color. Okay, so this is the core. Now depending on the various substitution, there is a different name of the structure. So this is the first one. So there is a various role of this rings. Okay, and if there is another, so this is the ring number A, which is the auxiliary ion ring. Each interactions with the peptidal transference center or PTC center of the 50th subunit. This B ring, this is side chain, this in the side chain in red, it is hydrogen bond with the phosphate group on and residue in the upper part of PTC. Okay, ring B, which is in the pink one, it is interaction with the PTC throughout the pi stacking. Then we have a ring C, which is present in green one. So this is the modulation of the pharmacokinetics, and ring D, which is in the black one, it may present in some auxiliary ion antibiotics, which is due to the improvement of lipophilicity. So the basic structure is ring A, ring B, and the side chain, and based on the improvement of the action and the lipophilicity and pharmacokinetics, ring C and ring D ring or chain D can be introduced, okay, to improve the pharmacokinetics of the molecule. So remember, the auxiliary ion works by the inhibiting the bacterial protein synthesis by binding to the 50th ribosomal subunit near the surface of 30th. Thus the formation of initiation complex is prevented. Okay. And one very important example of auxiliary ion class of antibiotic is your linazoloid. This is an antibacterial agent. So now moving forward, problem number 14 says identify the incorrect statement about mitomycin C. The options are: number A, it contains an aziridine ring; number B, it is not a prodrug; number C, it is the first step of its method of action is the reduction by NADPH; number D, it forms a indole-hydroquinone intermediate. So what is the incorrect statement about mitomycin C? Number A, it contains an aziridine ring; number B, it is not a prodrug; number C, the first step for the method of action is the reduction by NADPH, and number D, it forms the indole-hydroquinone intermediate. Identify the incorrect statement about mitomycin C. Number A, it contains an aziridine ring; number B, it is not a prodrug; number C, the first step of its method of action is reduction by NADPH, and number D, it forms a indole-hydroquinone intermediate. Let me check the chat box. Number B. So number B is the most given answer. Okay, let me check, and then let us discuss about this mitomycin C. So the incorrect statement about mitomycin C is, yes, number B, it is not a prodrug. It is the incorrect statement because mitomycin C is a prodrug. So this is the mitomycin C. Okay, this is the structure of it contains a aziridine ring, and the first step of the method of action is the reduction by NADPH, okay, and on CYP 450 reductase. And what happens, this mitomycin ring, after reduction it forms this mitomycin hydroquinone, and after this mitomycin hydroquinone is there is elimination of methanol, and we have this elimination happens from this position, and we have this indole-quinone intermediate, okay, indole-hydroquinone intermediate, and then in DNA what happens, this during the first step of the reduction, there is a radical formation and there is a Fenton reaction going on, which generates the cytotoxic hydroxyl radical, and because of this mitomycin DNA adduct forms and there is a mitomycin DNA adduct is formation happens, okay, and which is cross linked with DNA. Okay. So, the incorrect statement was number B, which is all of you have given the correct answer for this question. Okay. So, now problem number 15, it says that correct set of 3 coenzyme involved in the biosynthesis of acetyl CoA are number A TPP FAD NADH, number B TPP lipoic acid and FAD, number C FAD NAD plus and lipoic acid, and number D lipoic acid NADH and TPP. What is the correct set of coenzymes involved in the biosynthesis of acetyl CoA? What is the correct state of 3 coenzyme involved in the biosynthesis of acetyl CoA? Number A TPP FAD NADH, number B TPP lipoic acid FAD, number C FAD NAD plus lipoic acid, and number D lipoic acid NADH and TPP. Let me check the chat box. Number D, number C. Okay. Let me check which one of the following is the correct one. Okay. So, the correct set of 3 coenzymes involved in the biosynthesis of acetyl CoA is number B TPP lipoic acid and FAD. Okay. So, the conversion of pyruvate to acetyl CoA. So, this is the conversion of pyruvate to acetyl CoA. Okay. So, net reaction, it is the oxidative decarboxylation of pyruvate. The first carbons of glucose to be fully oxidized. Okay. The catalyzed by PDH, which requires 5 coenzymes TPP, lipoid and FAD are the prosthetic groups, NAD plus and your CoSH are the co-substrate. Okay. At the, so they derived from the TPP, which is thiamine, derived from TPP is thiamine B1, FAD is riboflavin, NAD niacin B3 and CoA is plantothanic acid. So, this is the pyruvate. So, we have this from pyruvate, we have this acetyl CoA, okay, and this the 3 set of your correct coenzymes is the TPP, lipoic acid and your FAD. So, as you can see, we have this this is the pyruvate dehydrogenase complex formation, okay, and then the delta G of this reaction is minus 33.4 kilojoule per mole. Okay. So, now next question, problem number 16 says which of the following statements is untrue regarding the cisplatin. Your options are number A chlorine groups are displaced before the drug becomes active, number B intrastrand cross linking of DNA results from action of the drug, number C base pairing of adenine and guanine is disrupted by this, and number D it has a square planar geometry. So, which of the following statement is untrue or which of the following statement is false regarding the cisplatin? Your options are number A chlorine groups are displaced before the drug becomes active, number B intrastrand cross linking of DNA results from the action of the drug, number C base pairing of adenine and guanine is disrupted by this, and number D it has a square planar geometry. Let me check the chat box now. Okay, number A is the most given answer. Let me check if number A is correct or not. So, the statement which is untrue regarding cisplatin is number C, base pairing of adenine and guanine is disrupted by this. It is the incorrect statement. So, this is the cisplatin, this is the structure of cisplatin and this is the structure of transplatin, which are the, so have the square planar geometry. So, what is the mechanism of action? First, what happened? There is a plasma membrane, okay. In the plasma membrane at the blood, the chlorine ion concentration is 104 millimolar, and into the cytoplasm the chlorine ion concentration is 4 to 10 millimolar. So, what happens? First, the chlorine ions are displaced and there is a and water is adds to the particular platinum, and we have this +2 charge here. Then cisplatin, it binds with the bind to the guanine, okay, and Cg base pair in the cytosine and guanine base pair, this particular cisplatin binds, not in the adenine and guanine, okay. And then there is a adenine and thymine base pairing. If you have checked, if you have read it carefully, you will identify this first statement is incorrect, because I have said base pairing of adenine guanine, but actually the base pairing happens in cytosine and guanine and thymine and adenine, okay. So, this Cg base pair, it is disrupted by the binding with this platinum of this particular cisplatin, and 1 to D CGP intestinal adduct which is formed, which is the major adduct. So, what happened? First, chlorine ions are displaced when it enters to the cytoplasm, and then it binds to the N7 position of the guanine, and this Cg base pairs are disrupted, and this is the mechanism of action, this interest and cross linking by which this particular cisplatin works, okay. So, statement number C was untrue regarding the cisplatin. So, now next problem number 17, it says which on the following statement is false regarding for secondary metabolites. Their options are: number A, they are derived from primary metabolites; number B, polyketides is one of the secondary metabolites; number C, they are directly involved in growth, development and reproduction; and number D, they are produced in small quantities. So, which of the following statement is false regarding the secondary metabolites? Number A, they are derived from primary metabolite; number B, polyketide are one class of secondary metabolites; number C, they are directly involved into the growth, development and reproduction; and number D, produced in small quantities. So, which of the following statement is false regarding secondary metabolite? Let me check the chat box and then I will discuss. Okay, so the answers which are given is number C, number C, number C. So, all of you think it is a number C. Okay, let me check if number C is the correct one. Yes, number C. They are directly involved in growth, development and reproduction is the false regarding the secondary metabolite. So, secondary metabolites are the compounds which are derived by the pathways from primary metabolic routes and are not essential to sustain the life of the cells. These compounds do not have any continuous production. Very often secondary metabolites are produced during non-growth phase of the cell. Secondary metabolites are the end product of primary metabolites such as alkaloids, phenolics, steroids, essential oils, lignin, resin and tannins. So, the differences between primary and secondary metabolites are: primary metabolites perform the physiological functions in the body like intrinsic functions. Secondary metabolites are derivatives of the primary metabolites. Primary metabolites directly involved in the growth, development and reproduction. Secondary metabolites play in ecological functions. Primary ones, they form during the growth phase of the due to the energy metabolism, and they formed near the stationary phase of the growth. Primary metabolites produced in large quantities and extraction is easy, whereas the secondary metabolites produced in small quantities and the extraction is difficult. The primary metabolites are same for all plants. The secondary metabolites are unique in different plant species. A part of the basic molecular structure of an organism is the primary metabolite, like ethanol, lactic acid, nucleotide, vitamins are the example of primary metabolite. But secondary metabolites are not a part of the basic molecular structure of organism. For example, pigments, antibiotics, drugs are the examples of the secondary metabolite. So, now I will move toward the next slide. So, problem number 18 says, which of the following is known as antigenic determinant? The options are: number A hapten, number B epitope, number C enzymes and number D paratope. So, which of the following is known as the antigenic determinant? Haptone, epitope, enzymes and paratopes. Which of the following is known as antigenic determinant? Hapten, epitope, enzymes or paratope? Let me check the chat box. Number D, number D, number C. Okay. Let me check if number C or D is correct or other two, any one of other two is correct one. So, the correct answer is number B, epitope. Epitope is known as the antigenic determinant. So, what are the haptens? Now, let us understand what are the each and every term. Hapten is a molecule too small to stimulate antibody formation by itself. However, when the hapten is combined with a larger carrier molecule, usually a serum protein, the hapten and its carrier together form a conjugate that can stimulate an immune system. Enzymes are an enzyme is a form antibody, and enzyme is often called the catalytic antibody, is a monoclonal antibody with catalytic activity. Okay, so there in enzyme we have a tumor binding site, and then we have a product activation site. Okay, and then we have the epitope, and epitope is also known as the antigenic determinant, is a part of an antigen that is recognized by the immune system, specifically antibodies B cell and T cells, and paratope is a part of antibody which recognizes the antigen binding site of the antibody, and a small region of 15 to 22 amino acid of the antibodies. Every region contains the parts of antibodies heavy and light chain. So, this is a antibody, and this is a paratope, and this is a antigen, and there is a epitope. So, antigen have epitope and antibody have paratope. Paratope and epitope, they fit with each other. So, we have this antigen antibody interactions. Okay, and so remember the antigenic determinant is the epitope which is present in the antigen, and it is a part of an antigen that is recognized by the immune system, specifically by antibodies B cell and T cells. Okay. So, now next problem. Problem number 19 says acyclovir, this structure is shown here, is an inhibitor of DNA polymerase selectively in virus infected cell. This is because number A, acyclovir first converted to monophosphate only by viral thymidine kinase. Number B, acyclovir does not enter infected cells. Number C, acyclovir first converted to monophosphate only by host thymidine kinase. and number D, acyclovir triphosphate is not accepted by host DNA polymerase. Read all the options carefully. Acyclovir is shown here. It is an inhibitor of DNA polymerase selectively in virus infected cells. This is because number A, acyclovir first converted to monophosphate only by viral thymidine kinase. Number B, acyclovir does not enter the infected cell. Number C, the acyclovir first converted to monophosphate only by the host thymidine kinase. And number D, acyclovir triphosphate is not accepted by host DNA polymerase. Which of the following is the correct option? Take 2 minutes and then I will show you the answer. Read all the options carefully and then write in the chat box. Let me check the chat box now. Yeah. Okay, so the answers are given number D, number C, number A. Okay, let me check which of the following is the correct one and then let us discuss about it. Okay, so the correct answer is number A, acyclovir is first converted to monophosphate only by viral thymidine kinase. So, this is if you have read the all options carefully, so I think you have figured out why it is the one. So, this is the acyclovir, it is a particularly it is a prodrug, okay, and this is the structure this given below, it is the acyclovir structure. So, this is a antiviral agent, this is a very famous antiviral agent. So, why this particular compound only inhibit the DNA polymerase in virus infected cells, but not of the human DNA polymerase, okay, for the human cells. So, whenever you are having any kind of DNA polymerase inhibitor or any kind of other enzymatic inhibitor like topoisomerase inhibitor or so on, and if you are you are targeting the protein inhibition, protein synthesis inhibition or DNA replication inhibition in particularly bacteria, virus or everything, first thing you have to take care of is that these particular compounds need to be selective. Means if you are targeting to inhibit the DNA polymerase of virus, that particular agent must not inhibit the DNA polymerase of the humans also. Okay, so there are various techniques or various ways by which we can do it. So, acyclovir for this particular compound, what happens, this compound is converted to monophosphate and this happens only by the viral thymidine kinase form, and that monophosphate form, it is the active form of this drug which exerts its antiviral activity by inhibiting the DNA polymerase, but this particular compound is not converted to the monophosphate by host thymidine, and that is why it does not affect the host DNA polymerase enzyme. Okay. So, now last question of today's session is number 20. It says that diminished delivery of oxygen to the tissues is termed as number A, hypoxia, number B, ischemia, number C, homeostasis and number D, metabolism. So, what is the diminished delivery of oxygen to tissues is termed as hypoxia, ischemia, homeostasis and metabolism. Now, let me check the chat box. So, the answers are given is number A. So, I have only number A as the answer. So, let me check if it is the correct one. Yeah. So, hypoxia is the term which is which means the diminished delivery of oxygen to the tissues. So, basically hypoxia is a condition in which the body is deprived of adequate oxygen supply. Okay. The altered brain, the brain sends the signal to the rest of the body whenever there is a hypoxia, and if there is a hypoxia means there is inadequate oxygen into the tissue in the tissues. So, what happens, our brain gives us the signal that there is the inadequate oxygen supply. So, which means we need to increase the oxygen supplies in the body. Okay. And this can be done by increased breathing rate and constructed vessels in the lungs, increasing the heart rate, and dilated peripheral blood vessels in the arm, legs and hands and feet. So, what is the term the term ischemia? It means it is a state when a tissue or a organ has its arterial perfusion lowered relative to the metabolic needs, or ischemia is defined as the condition inadequate blood supply to an area of tissue. It is an inadequate blood supply in the part of the body even the point complete deprivation. It is simply defined as a condition of inadequate blood supply. So, the hypoxia means inadequate oxygen supply, ischemia means inadequate blood supply. Now, homeostasis is a condition in which the internal environment of the body remains relatively constant despite changes of the external environment. Examples would be the maintenance of body temperature and the levels of glucose in the blood. And then we have metabolism. It is a definition, the sum of all chemical reactions in the body, anabolic versus catabolic reaction. Anabolic reaction use chemical energy to synthesize products, and catabolic reaction they break down substances to generate chemical energy. So, this is the all means this term. So, hypoxia, ischemia, homeostasis and metabolism, these terms are means I have mentioned whatever this means these terms are related to. So, this was the last question of today's session. So, what we did in today's class was we discussed about various agents like various anti-cancer agents, various antibacterial agents. We discussed their mechanism of actions, what are the structural features which are important for them. So, we discussed a lot of about this biological portions in today's class, and most of you have answers very well the questions. And also we discussed about little bit about antigen antibody, then we discussed about the your biological chemistries, and we discussed about the metabolites. We discussed about the mainly the various terms of metabolic which are important in the metabolism. So, today it was the week 4 live session of this NPTEL course of medicinal chemistry, and we discussed about the week 5 session. So, I hope this problem solving session will be helpful for your solving the assignment portion. So, now in the chat box I am sharing my email ID. Okay. So, this is my email ID. If you have any doubt, any query, any problem regarding today's class and any time, not only today's class, if you have any doubt, any query in previous classes also, the week 1, 2 and 3 also, you just can post your query or your doubt in this email ID. Okay, there is a discussion forum on the SWAM portal also. So, if you have any problem, any doubt, you can also just go to the SWAM portal and you can just have this look at these doubts. And I think all of you have the access with this problem solving session videos and problem solving session PPTs, right. So, if you go to the SWAM portal, just let me show you once this thing. So, because one student was asking me about the videos and the lecture PPT. So, let me just show you how this thing works like. So, yeah, not medicinal, yeah. So, what you have to do is you have to just go to the SWAM portal. I think all of you have registered in the SWAM portal and you have to log into the SWAM portal, and there you will find there is a particular portion which is called the problem solving session, and in that, so you have first you have to log into the SWAM portal. Just let me show you where you have to find out this particular problem solving session video and PPT, because most of you sometime mail me about the PPT, but you just here. If you open this problem, this is SWAM portal and this is the announcement pages of this medicinal chemistry course. Okay, and this is the medicinal chemistry course. Here you will find the link for the class and also the timing of the class, and if you click on this portion, you will have the problem solving session, and if you click on this, you will get the problem solving session lectures and also the videos. Okay, so here there is a lectures and videos are present here, so you can find it out from this SWAM portal, the problem solving session. In case you do not find it, so you just can let me know. Okay. So, now I will close the session for today. I hope the session will be helpful for you for solving your assignment and everything. Okay, and let me know if you have any doubt or any problem. This was the week 4 live session for NPTEL course of medicinal chemistry. Next week we will have another problem solving session on Tuesday from 6 p.m. to 8 p.m. and that will be our week 5 live session. So, please join on time, and if you have any problem, feel free to reach me. Okay. So, thank you all for joining, and I will close the session for today.